Hello everyone. Today we will be exploring about cell biology in environmental detection. I am Dr. Shrisha Naik and we will be exploring how the pollutant and the cells interact with one another. The cell biology is a specific branch of biology surrounded by structural and functional aspects of cell and their component. It is being effectively used as an important parameter for screening of environmental mutagen and carcinogen. So cells, what happens is when in biology, whatever mutagen, have to come in contact with the cells, and their interaction is what we are looking for in the detection of mutagens and carcinogens in the environment. So they act as, let us explore how they are able to help us. So they cell as a cellular target. So what does that mean? So this, as the cell interact with one another, so there are lot of DNA damage or the cellular damage that is going on. So the cells are primarily sites of interaction with toxins, marking cell biological methods valuable for assessing cellular damage. And also they act as dosimeter. So dosimeter, what do you mean by that? So biological dosimeter is we will be able to quantify the extent of DNA damage caused by environmental toxins. So let us see what are the cellular targets and how these cellular targets are useful in exploring the environmental toxins. So we have cell membrane, cell organelles, and we have chromosome. So cell membrane is the first line of defense against cell toxins, and it is integral in the crucial for the cell survival. So we have in the cell organelles, we have mitochondria, nucleus, lysosomes are the susceptible for damage against the environmental toxin. Chromosome, which is the DNA genetic material, or it is what all the cells have, and all the information regarding the cellular life is contained in the chromosome, and it contains DNA in it. So chromosomes carry genetic material, are particularly vulnerable to damage, leading to the mutation and potential health risk. So these are some of the targets that we have that we are seeing. So what are the organisms that we are looking into? So one of the organism and the important organism is microorganism. So these small microorganisms such as bacteria, yeast, and are often used in genetic damage assessment due to their rapid growth and ease in manipulation. So they are very easy to be cultured. You can take them in the petri dish and you can culture them. So once you culture them, they are easy to be manipulated. So then we have plants. So plants with their diverse genetic material provide valuable models for studying of effect of environmental toxin on genetic integration. So plant is also a very good indicator. So presence of certain plants in an environmental condition where there is high concentration of pollution is an indicator for lot of pollution that has been going on. So then we have laboratory animals. So there are several different laboratory animals that are used for the detection of pollution. Example is mice. You have rats, guinea pigs. So these are some of the animals that are used to detect the pollution. There are so many other examples that are used for detection of pollution. So some of the common ones are these animals. Then we have human cell culture. So human cell culture provides a very good model or an indicator of cellular damage and the DNA damage that is going on inside the cell, because you can culture what is being happening to the human being, how these toxins affect the human cells can be easily cultured and detected and can be analyzed using human cell culture. So these are some of the tests that we will be looking for. So one of the important tests that we will be seeing is Ames test, which is widely used in the screening So we have Salmonella typhimurium. So Salmonella typhimurium is a bacteria that causes typhoid. So it is one of the strain of typhoid bacteria that is used to detect the chemical mutagen. So this type of Salmonella typhimurium is susceptible for mutagenic changes. So a small concentration of chemical mutagen that is added into the culture media can be detected using Salmonella typhimurium because they are susceptible for mutagenic changes. So how they are detected is the test measures the frequency of reversion of mutation at the histamine locus. So there is an protein and there is an enzyme that codes for histamine. So detection of mutagenic potential of the chemical is done by reversion of mutation. So the mutation, that is auxiotrope, histamine auxiotrope, will be reverting to the plus and minus activity of the histamine will be used to detect the mutagen in the environment. So this is one of the important tests. So then we have different test, that is cytogenic assay to detect chromosome damage. So what are those? So there is, you have chromosomal aberration. So the mutagen also interact with chromosome. So cytogenic assays assess the extent of chromosome damage, including fragmentation, bridge formation, disruption of cell division. So the mutagen, they will interact with the cell and they will stop the chromosomal division during the mitotic or meiotic division. So if it is an a somatic cell, it is mitosis. If it is germinal cell, it is meiosis. So they have chromosomal changes can occur. So that will lead to fragmentation of DNA and the formation of different bridges and disruption of cell division. So cell won't divide further. So then we have other method of testing is you have micronuclear test. So the interaction of the chemical mutagen or the toxin, what will causes is the makes the nucleus small. So these damages in the nucleus make causes the repair enzyme to be released, and some of the cells are the some of the cell will have very small nuclei, and there will be large scale chromosomal fragmentation. So the size of the nucleus is a very good indicator to analyze the extent of chromosomal damage. Then is formation of sister chromatid. Sister chromatid exchange detects the damage of DNA molecule and mixed exchange of chromosomal segment. Okay, so let us explore in the next slide. We'll explore the different aspects of chemical mutagen and in its environmental detection. Thank you.